Hello everyone, I am Nusayba Uthman Al-Damouk, a third-year pharmacy student from Ain University. Today we will be presenting to you our project about the promising use of NONO Inhibitor as Anti-lung Cancer Therapeutic Agent. Lung cancer is the second most common type of cancer and the leading cause of cancer-related deaths, according to the World Health Organization as of 2020. Despite the available therapeutic plans, treatment outcomes are still suboptimal and patients are at high risk of death. Thereby, development of drugs aiming at novel targets with promising results for cancer treatment is urgently required. NONO is one such target that has been suggested by multiple previous studies. NONO, or non-POU domain containing octamer binding protein, is a protein also known as 5,4-KD nuclear RNA and DNA binding protein, abbreviated as P54NRB, that belongs to the multifunctional Drosophila behavior human splicing family of proteins. It has a variety of functions, including cell proliferation, apoptosis, cell migration, and DNA damage repair. Furthermore, NONO plays a role in cell attachment to the extracellular matrix components, where its inhibition enhances the cell attachment and significantly reduces cell migration. With that being said, it seems very reasonable to target and inhibit NONO, which has been shown to be overexpressed in a variety of types of cancers. It should be noted that NONO activation requires structural changes via post-translational modifications like methylation and phosphorylation accomplished by protein arginine methyltransferase, abbreviated as PRMT, and cyclin-dependent kinase, abbreviated as CDK, respectively. The aim of our study was to assess the role of NONO in proliferation and metastasis by inhibiting the methylation and phosphorylation of NONO, thus inhibiting its activation. This was accomplished by blocking arginine methylation and threonine phosphorylation by inhibiting the PRMT1 and 4 and CDK1, and the effects were tested on two lung cancer cell lines, which are the A549 and H292. Cell proliferation percentage and cell viability percentage of both cell lines were measured following the inhibition of PRMT1 and 4 using ADOX at a concentration of 20 micromolar. Cell proliferation percentage and viability percentage of both cell lines were also measured following the inhibition of CDK using roscovitin, which is a CDK1 inhibitor, at a concentration of 12 micromolar, and the cell lines were incorporated in a DMSO vehicle that was used as the control for this experiment. Cell viability was calculated through the use of a hemocytometer, where the living cells were viewed as clear and colorless, while the dead cells had a blue color due to the triband blue dye that was employed. After the number of live cells and dead cells was calculated, the sum of their amounts was calculated, and the live cell count was divided by the total cell count and multiplied by 100 to calculate the percentage viability. Cell proliferation was calculated by comparing the cell count of the treated cell culture and the control cell cultures, and the corresponding percentage was then calculated.
Thank you, Nusayba. My name is Khawla Abu Hayat, a fourth-year student at Ain University, and I will be speaking briefly about the results of our experiment. The results obtained following administration of ADOX to A549 and H292 cell lines are as follows. Cell proliferation of A549 cell line decreased to 63.8% after 48 hours following ADOX administration. Meanwhile, cell viability of the same cell line was reduced to 53% after 48 hours post-ADOX administration. In regards to cell line H292, the cell proliferation was effectively reduced to 65.1% 48 hours post-ADOX administration, while cell viability decreased to 65.9%. The results obtained after administering roscovitin to A549 and H292 cell lines are as follows. Cell proliferation of A549 cell line decreased to 40% after 96 hours following roscovitin administration. Meanwhile, cell viability of the same cell line was reduced to 42.9% after 96 hours post-roscovitin administration. In regards to cell line H292, the cell proliferation was effectively reduced to 33.9% 96 hours post-roscovitin administration, while cell viability decreased to 30%. Now, from the graphs, we can observe some general trends. For example, The addition of ADOX to the A549 and H292 cell lines resulted in a significant decrease in cell proliferation and viability. The decrease was more prominent in A549 cell line than in H292 cell line. The addition of roscovitin to cell lines A549 and H292 also resulted in a steep decrease in cell proliferation and cell viability. Contrary to ADOX, roscovitin caused a more prominent effect on H292 than A549 cell line. The vehicle in which the cell lines were incorporated, DMSO, had negligible effect on cell proliferation and viability. Overall, roscovitin resulted in a greater decrease in cell proliferation and viability compared to ADOX.
Hello everyone, I am Fatemeh Abdi, fourth year student of pharmacy in Ain University, Abu Dhabi campus. The results from this experiment have shown that NONO plays important role in human tumorigenesis by having effect on the cell proliferation and viability. PRMT14 by binding to NONO methylated, while CDKs by binding to NONO will phosphorylated, which results in the activation of the NONO and subsequently proliferation and metastasis. For this reason, blocking the arginine methylation and threonine phosphorylation via inhibition of the PRMT14 and CDKs has been tested on 2 lung cancer cell lines, which we use A549 and H292 in order to inhibit the proliferation and metastasis. Inhibition of the PRMT14 has been done via ADOX, and inhibition of the CDK1 has been done via roscovitin. The correlation between inhibition of NONO and the amount of reduction of the cell proliferation and cell viability has been analyzed. On the A549 cell line, by addition of the ADOX, we had reduction in cell proliferation by 64% and 53% for the cell viability. These amounts with the roscovitin were 40 and 43% respectively. On the H292 cell line, by addition of the ADOX, we had 65% for the cell proliferation and 66% for the cell viability. By roscovitin, we had 34 and 30% reduction in the cell proliferation and cell viability respectively. Our findings reveal a link between the methylation and phosphorylation inhibition and considerable reduction in the cell proliferation and viability. However, the efficiency differed across the A549 and H292 cell lines. Our results indicated PRMT14 and CDK inhibitor and subsequently NONO inhibitors as a promising candidates that can be effectively work as an anti-lung cancer therapeutic agents. This strategy has the potential to develop further in the future and opens the possibility of establishing new class of drugs for the treatment of the lung cancer. Better understanding of context of NONO inhibition itself and evaluation of its effect on the cancer cells and testing on the cell proliferation and viability will make it more therapeutically valuable. Also, analysis of the amounts of methyl NONO and phospho-NONO in cancer cells in comparison with the normal cells can clarify the concept more. We suggest further research on evaluation of the quantities of the methyl NONO following the inhibition of the PRMT, as well as the evaluation of the prevalence of the phosphor NONO following the inhibition of CDKs.